Hello, welcome to Goal 2, Module 2. Let's talk about bonding. This module, the second one for Goal 2, will cover the basics of bonding and how that bonding contributes to the qualities of water and the differences between acids, bases, and salts. The objectives are to distinguish between ionic, covalent, and hydrogen bonds, explain how the molecular structure of water relates it to its important properties in the human body, and distinguish between acids, bases, and salts. Electrons are arranged in shells with 2, 8, and 8 electrons in the first 3 shells respectively. Atoms are happiest when these shells are filled. This rule is key when it comes to how atoms bond with each other. Rarely are elements hanging out all by themselves. They connect or bond with other atoms, with each other, like 2 oxygen molecules like to hang out together, or with different elements, like 2 hydrogen and 1 oxygen hang out to form water. The 3 main ways that atoms interact or bond are ionic, covalent, and hydrogen bonds. These 3 types of bonds make up all the different molecules in your body. The first one we're going to talk about is ionic bonds. Ionic bonds form between atoms that have just 1 or 2 electrons in their outer shell, or 6 or 7 in their outer shell. They want to be full, or they want to be empty, so the next lower shell is full. So they tend to donate 1 or 2 or receive 1 or 2 to fulfill the 2-8-8 rule. In the diagram shown here, sodium has 1 electron in its outer shell, and if it's lost, then its outer shell number 2 would have 8, and it would be happier. Chloride has 7 in its outermost shell and would really like 1 more to be happier. This is exactly what happens. Sodium loses 1 electron and donates it to chloride, which receives it. Because sodium has lost an electron, it's now positively charged. Remember, it still has the same number of protons. And chloride is now negatively charged because it has, I'll go back to the previous slide here. Because it has 7 in its outermost shell, and now it has 8, and it's much happier. Because it's now negatively charged with that extra electron, the 2 atoms or elements tend to hang out together because one is positively charged and one is negative. If you put any ionic compound in water, the ions or electrolytes will separate. This is called dissociation. They float around in the water, but the water molecules kind of get between the ions and pull them a little further apart than they would be if they're not in the water. The ions or electrolytes will still be charged because the electrons in the solution are still either an extra one is on chlorine or sodium is missing one, and therefore the solution will conduct electricity. You will be testing conductivity of various solutions in lab. Solutions that have more ions will conduct more electricity. The second type of bond is much stronger. This bond, the covalent bond, again is created to fulfill the 2-8-8 rule. But instead of donating and receiving, the electrons are shared between 2 atoms. Because losing or gaining 3 or more electrons is just not probable, the sharing keeps the 2-8-8 rule possible in this type of bond. Notice in the diagram that 2 hydrogen atoms, which each have 1 electron, can get really close together and share both. Think of those speeding around in a figure-8 pattern around the nuclei of both atoms. Essentially, the electrons are around both atoms all the time. Remember, they're going at the speed of light. This means they each have 2, fulfilling the 2-8-8 rule, and it is much more stable than being alone with just 1 electron. When electrons are shared, this forms a covalent bond. Generally, a little bit of energy is required to make them, and a little bit of energy is released when they break. They do not dissociate in water like the ionic bond. It's too strong for water to get in between the atoms. Because covalent bonds store energy, or are potential energy, because if you break them you can get a little energy, when we need energy, we need to eat covalently bonded molecules. Carbohydrates, fats, and proteins are all made of covalently bonded molecules. Glucose, fatty acids, and amino acids, the small building blocks of those macromolecules, are covalently bonded, and the more we eat, the more energy we get. We may store some of this energy, but breaking these bonds is what generates energy for our body cells to do their jobs. These two diagrams show the difference between covalent and ionic bonds. Notice the shared electrons, the covalent bond, the atoms are much closer together than in an ionic bond where electrons are donated by one and received by the other. Atoms can share one pair of electrons to form a single covalent bond, but some atoms share two or more pair. When you share two pair of electrons, so 4 electrons, that is referred to as a double covalent bond. If you share 3 pairs of electrons, or 6 total electrons, that is a triple covalent bond. Carbon dioxide has 2 double bonds between the carbon and the oxygen on one side and the oxygen on the other. Nitrogen, that's in air, is actually N2. It's a triple bond between the 2 nitrogen atoms. Remember, a covalent bond breaking usually releases a little energy, so a double or a triple bond breaking would release even more energy. However, the human body has a tough time breaking double or triple bonds. Often we cannot do that. Carbon dioxide has 2 double bonds, as seen in the diagram, and we cannot break that down any further. Therefore, we exhale it. It is completely useless to us. Only plants can break the carbon dioxide bond, this double covalent bond, and use it to make sugar and oxygen, which we love. Sometimes electrons are not shared equally between 2 atoms. If they are equally shared or pulled between the 2 atoms equally, this covalent bond is said to be nonpolar. Think about a tug of war. If the rope is straight across from the center point and both people on either side have equal muscles, they're pulling equally. That's a nonpolar bond. However, if the polar sharing is not quite equal, the bond is said to be polar, because one pole gets a little more of the electrons than the other. Water is a polar covalent bond because the hydrogen atoms are bent, not equally pulling on the oxygen, making the hydrogen slightly more positive and the oxygen slightly more negative. Okay. Methane is nonpolar because the 4 hydrogens are equally pulling on the carbon. While it's not always important that you know if something is polar or nonpolar, generally polar molecules like to hang out with other polar molecules, and nonpolar tends to hang out with nonpolar. Like likes like. This is why oil, which is nonpolar, doesn't really like to hang out with water. If you put oil and water together and shake them up, as soon as you stop shaking, they separate right back into the water and the oil, or the polar versus the nonpolar. Here's an example of polar and nonpolar molecules robbed from the web. Water is polar. Sometimes other polar molecules, because they dissolve in water, are referred to as hydrophilic, which means they love water. Substances which dissolve in water are hydrophilic. Fats and oils, which are nonpolar, are sometimes referred to as hydrophobic because they fear water or they don't like to dissolve in water. The last type of bonding is a bond that is between 2 different molecules or between 2 parts of a very large molecule. This bond, the hydrogen bond, is a very weak bond, and it always is between a hydrogen and an oxygen, or a hydrogen and a nitrogen. Okay? Because it's so weak, it's easily broken by heat or a change in pH. Our proteins in our bodies fold up because of weak hydrogen bonds between the hydrogen and nitrogen, or hydrogen and oxygen, along that molecule. And if they don't fold up properly, they don't work. Perhaps you've heard of protein folding, or unfolding, or denaturing. It is a very active area of research. DNA's helical structure is also because of hydrogen bonds. Without the hydrogen bonds, DNA doesn't work either. Water is especially affected by hydrogen bonds. When you have water on a spoon and you turn the spoon over, all but one drip of water will fall off the spoon because of gravity. But that last drip has enough hold in it due to the hydrogen bonds between the water molecules to keep it there, even though gravity is still pulling down. Surface tension of water is due to these hydrogen bonds, and that's what keeps that last drip on the spoon unless you flick it off. This is also why insects can walk on water, because between the molecules of water there are hydrogen bonds, and insects aren't heavy enough to break it. There is a weak attraction of the hydrogens of one water molecule and the oxygen in the second molecule. You can see that in the diagram here, the little dashed lines. Notice the white balls, which are the hydrogen, are slightly attracted to the red or the oxygen molecule of a different water molecule. Both of these images show some hydrogen bonds between either the molecule of water up here or within a protein. And again, proteins do not fold up unless their hydrogen bonds have formed. And heat or acid or base can break those hydrogen bonds. This is why we don't want to get too hot or be in acidosis or alkalosis. Our proteins unfold, then they don't function, then we die. Water is wonderful stuff. Because of its polarity, lots of things will dissolve in it, and therefore it's both a lubricant and a solvent. Because of hydrogen bonds, ionic compounds dissociate in it, and therefore electrolytes are available to our cells, which are bathed in a fluid that is mostly water. Blood, lymph fluid, and interstitial fluid is almost all water, so ionically bonded compounds will dissociate, and those electrons can then be taken up by our cells. Sugar and amino acids, or proteins, also dissolve in water, as does DNA. Only lipids, fats, and oils do not. But since our cell membranes are made of these, the lipid membrane can keep the cell's water apart from the water outside the cell, and fats can diffuse in. Water also has a high specific heat content. It holds heat and takes a lot of energy to warm it up. The oceans soak up a lot of heat, but so does our blood, protecting us from rapid heat loss during times when it's colder than body temperature in the room we're in or outside. As just stated, ionic compounds come apart or dissociate in water. If so, you get electrolytes. If one of those electrolytes happens to be the hydrogen ion, that's the H plus right here is the hydrogen ion, then the solution will become acidic. If one of those ions or electrolytes happens to be the hydroxyl ion, OH negative, the solution becomes basic. If neither one of the electrolytes are around, OH negative or H plus positive, then the solution is considered a salt and the pH doesn't change. pH is essentially how much hydrogen ion is in that solution. The more the hydrogen ion, the more acidic. The fewer, the less acidic or more basic. This is a general diagram of the pH scale, which goes from 0 to 14, 0 being very acidic, 14 being very basic or alkaline. Generally, human tissues really like it about neutral, which is 7. Our blood pH is 7.4, and if it varies very little from that, our tissues are damaged, proteins unfold because of the hydrogen bonds breaking, and we are in a world of hurt. If you look at this pH scale, we tend to like things that are acidic. We call them sour, but orange juice, soda, lemon juice, tomato juice are acidic foods, and we can eat those. If you go up above 7, we don't eat too many things up here. It tastes kind of nasty. Milk of magnesia or Pepto-Bismol is very basic. Milk is pH 7.5, and that's about as basic as we like food. Again, after dissociation, and remember only ionic compounds dissociate, electrolytes are generated. If one of those electrolytes is the hydrogen ion. That solution is an acid. If one of them is OH, the OH part won't come apart because it's covalently bonded, but the OH together, this ion or electrolyte, makes the solution basic. If neither one are present, the pH doesn't change and the solution is considered a salt solution. So how do you figure this out? The first question you need to ask yourself is, when you're looking at a molecule like NaCl or HCl, determine if it's ionic or not. One of the easiest ways to tell if it's ionic is covalent molecules in biology tend to have carbon, oxygen, or nitrogen in it. If it doesn't have any one of those three, it's usually ionic. If it is ionic, go to number 2. It's going to dissociate in water and write out the electrolytes or ions that are formed. If one of those ions is H+, it's an acid. If not, look at question 3. Is it OH? If there's an OH negative present, it's a base. If not, it didn't have either one, it's a salt, and the pH did not change. We're going to do an electrolyte lab in class, and hopefully you'll get more of that during that lab. So looking at these molecules, you should be able to determine if they're ionic or covalent, and if they're ionic, you should be able to determine if it's an acid, base, or salt. A couple of things to remember. OH is covalent all by itself, so it doesn't come apart further. But OH negative is an ion and makes a solution basic. Molecules that have carbon, oxygen, or nitrogen are usually covalently bound to whatever else they're next to, unless an H is first and followed by more than one element. Then the H's are ionically bonded and it's usually an acid. Okay. The next slide shows the answers to each one of these. So you can pause the slideshow and see if you can figure them out, and then continue to see the answers. So here are the answers. You can look these over after you've worked them out. Last but not least, don't forget your online lesson, your lab, your online textbook, and the internet are great resources to help you find more information. Use your periodic table. Don't be afraid to ask questions.